Hello everyone, my name is Nicholas, and today we will be discussing future treatments of Duchenne's muscular dystrophy. Our first treatment being discussed today is transplicing. However, before we get into what exactly that is, I'd first like to give a quick refresher on dystrophin and Duchenne's muscular dystrophy. Dystrophin is the protein that's rendered non-functional in DMD. Dystrophin is one of the largest genes found in the human genome and thus has many sequences that can potentially be disrupted, giving rise to a non-functional product in the DMD phenotype. Under normal circumstances, when dystrophin is properly translated, it is then localized in the dystroglycan complex. The DGC for short is critical for the muscle cells to maintain their health because it preserves the sarcolemma membrane as well as allows them to communicate certain signals between their intracellular and extracellular environments. If we take a closer look at the dystrophin mRNA transcript, we can then simulate certain events in which dystrophin has been rendered non-functional, or even in some cases where its function has been semi-restored. The first portion of this figure shows a scenario in which dystrophin is normal. This is represented by the lack of gaps between the exons despite their unique shapes. The second portion of the figure shows a scenario in which dystrophin has been rendered non-functional. This is because exon 50 has been deleted and thus left exon 49 to border exon 51. Exon 49 and 51 do not have compatible ends, which then is indicative of a frameshift mutation, thus leading to a non-functional protein and the DMD phenotype. The third portion of this figure shows a scenario in which the function of dystrophin has been restored. This is because not only exon 50 has been deleted, but also exon 51. This then leaves 49 to border 52, which are compatible in ends. This gives rise to a truncated protein that is semi-functional and results in a lesser severe phenotype known as Beckerman muscular dystrophy. This phenomenon has caused scientists to search for a way to replicate this scenario to offer a potential treatment for those with certain types of Duchenne's muscular dystrophy. This type of therapy is known as exon skipping therapy. One example of exon skipping therapy is transcriptional exon skipping. This is in which drugs cause RNA polymerase to skip over certain exons in order to restore their reading frame. Another approach is alternative splicing exon skipping, in which drugs are given in order to cause the spliceosome to splice out undesired exons in order to restore the reading frame. The pros of these types of therapies is that they can be delivered via drugs and result in a lesser severe dystrophy known as Beckerman's muscular dystrophy. The cons are that they are specific to only certain exons and cannot restore full dystrophin function. This is where transplicing comes in. Transplicing is another form of exon skipping therapy. However, instead of looking to focus on one transcript, it focuses on two. So splicing in its traditional sense is known as cis splicing, in which exons are joined together ultimately from the same transcript. Transplicing involves another transcript in which an exon from another transcript is taken and then supplemented for the final product. This is seen on the right hand side, in which the red exon is then incorporated into the final product. In the case of Duchenne's muscular dystrophy, transplicing can then be used to treat it, as shown as this experiment from back in 2013. In this experiment, scientists took an MDX dystrophin mouse, which has a mutation in exon 23. They then supplemented the MDX mouse with a transcript that has a normal functional exon 23. What they found was that the exon 23 from the supplemented transcript was then able to be incorporated into the final product, giving rise to a functional dystrophin in the end. This is then visualized in the next figure, in which they stained for functional dystrophin in their mice treated with this PTM. The top three figures of this figure show the controls. The first being a mouse with normal dystrophin function. The second with MDX, so should have little to no expression of functional dystrophin. The third with a mutation in the different portion of the dystrophin gene. The bottom two, MDX plus PTM and MDX4CV plus PTM, then show what happens when they are supplemented with that second transcript. You then see the visualization of dystrophin showing that the product is actually functional. The pros of this approach is that it can restore dystrophin in its full function, not just partially. However, in order to do this, it requires stable genomic integration. However, compared to other future therapies such as genomic repair, it does not require precise genomic integration, as it does not need to edit the dystrophin gene itself, only supplement it. Another con, however, is that it cannot be generalized like a drug delivery treatment, as it would probably need for individuals to give their cells in order to be treated and then transplanted back in. However, another pro is that this therapy could then potentially be supplemented with other therapies such as exon skipping therapy, because exon skipping therapy can then be used to make certain spike sites less effective, thus making the supplement and transpicing more effective.
So now we're going to be looking at andosiloxanide, which is different than the drug type of therapy that Nick was talking about, which was more focused on exon skipping and more on the genetic component. This focuses on the inflammation and pathway within exon or within muscular dystrophy. So what is it? So this drug contains two components, salicylic acid and fatty acid. So salicylic acid has been implicated in counteracting inflammation, and so has fatty acids. However, they've never been combined together, and thus they may produce some important benefits that are not available in independent drugs. So endosiloxanide is a combination drug therapy that contains both salicylic acid and the fatty acids, and this helps to create this a better drug therapy. So how does this drug really work? So well, you have this DMD mechanism that we were going over with, where this DMD mechanism is lacking after when dystrophin is not present, then the DGC will not form and lead to a problem with the mechanism that helps to retain muscle rigidity. So you have a muscle where it's continuously inflamed and irritated, and the nerve that's continually irritated, and that's just a representation due to the fact that there's inflammation that's happening within this pathway. So if you look at the normal biceps versus the muscular dystrophy, you will see that the normal biceps are a lot bigger compared to the muscular dystrophy, which is something that we need to address, and that's due to weaker inflammation. A good example is a border. So this is a border, and you have your body, which is on the inside inside of the border, and then the outside of the body. So that represents our body right now. So if there's an explosion, there's going to create a hole within the border, and soldiers will try to come in. And these are foreign pathogens, and the only way to attack them is to create specific defenses to these soldiers, essentially, and try to help prevent and help the immune system react to these soldiers in a specific manner. So this pathway that we are going to be looking at is the NFKB pathway. And this pathway is important because it's implicated in the inflammatory response within the body, and is when it's continuously activated, will lead to muscular dystrophy. We'll be focusing on the P50 and this trimer area, and we're trying to block it because these components are important for making sure that by downregulating or upregulating these components, we can ensure that we have a better control of the inflammation pathway. So this drug therapy is in clinical trials right now, and it represents something that's right around the cusp and is more general. And considering that the drug that we were talking about is more specific to therapy, this is something that's going to be a lot more general and more accessible to the population, because this is not just for DMD but other types of dystrophies. However, that being said, the con of this is that because it's not specific, there are other side effects that may result. However, preliminary testing in the phase 1 results have shown that this drug does not have that many safety issues as other drugs, and can be a good candidate for drug therapy. However, the future of the drugs are going to be something that's going to combine both of these.